Hi everyone. Today we're going to be talking about neuromuscular physiology. We're going to be talking about how the nervous system and muscular system work with one another. We're going to discuss how signals from our nervous system cause muscular contraction, because obviously if we didn't have our nervous system, our muscle tissues would not just respond on themselves, so we need to communicate with the nervous system. We'll talk a little bit about how cells receive and transmit signals. We're going to talk about the contractile machinery within the cells. This is referencing our actin-myosin cross-bridge cycling, as well as troponin, tropomyosin, calcium, all that good stuff, as well as we'll go through the muscular contraction steps, and then how we also recruit muscle fibers. Okay, so starting off, when we talk about neuromuscular physiology, we have to talk about the term excitability. Electrochemical signals, ultimately what that means is the transmission of action potentials. And the ability of the tissues to generate these electrochemical signals or these action potentials gives them the characteristic of excitability, meaning capable of responding to a stimulus. So excitability ultimately means capable of responding to a stimulus. Two things must be present if, in fact, there is going to be excitability, if these tissues are allowed to send these electrochemical signals. One, the sodium-potassium exchange pump that is constantly operating. Two, must be a greater membrane permeability to potassium than sodium. Those two characteristics give the tissue the polarized characteristic. If a cell is polarized, it is more negative on the inside relative to the outside. That's very important to know. So those two characteristics, once again, are that the sodium-potassium exchange pump is constantly operating. Two, there must be a greater membrane permeability to potassium than to sodium. Those two characteristics give us the tissue the polarized characteristic. And if a cell is polarized, that means it's more negative on the inside of the cell relative to the outside of the cell. Okay, so when we talk about depolarization, we're referencing the fact that the cell is going from a negative state to a positive state. And that's how we generate action potentials or the transmission of those electrical signals from one cell to another. That's how we're going to communicate. So there are different stimuli that can alter this positive-negative relationship. If the stimulus is of threshold intensity and the inside negativity of the cell is altered, then that cell becomes depolarized. That's the key occurrence that allows an action potential to be propagated from one point to the other. Depolarization must precede the movement of an action potential. So ultimately, depolarization is causing this action potential, so causing the signal going from one cell to the other. Okay, so we're going to talk about neurotrophic chemicals. These are the neurotransmitters, and the nervous system is going to be using them. They'll only work if we send them from the presynaptic side to the postsynaptic side. These chemicals alter the membrane permeability on the postsynaptic side. If you alter the permeability enough, you can create depolarization and then an action potential within that cell. So that's ultimately what we're trying to do. We're trying to go down the axon over here into the terminal bulb, which is part of the nervous system. And what will occur here is we're going to go over these steps more in depth in just a few minutes. But ultimately, we're going to be releasing acetylcholine, and they'll be attaching to alpha receptors over here, and we want to alter the membrane permeability on this side, so then an action potential can propagate down within the muscle cell, which is going to then signal muscular contraction. So this is how ultimately we're getting to get down to the muscle cell level in order for us to contract. So if you alter the permeability enough, you can create depolarization on this side. The location where this occurs is called a synapse. So this is a synapse right here. In between, this is our nervous system, so this is our axon terminal, and then this is our muscle cell right here. But there is a space in between the two. The nervous system and the muscle cell are directly not touching one another. There is a space here, and that's called the synapse, or the synaptic cleft. We can state that threshold has been attained if, in fact, the membrane permeability on the postsynaptic side has changed enough that we have caused a depolarization. Here's a larger version of the diagram for you to see. So here we have the axon terminal, and then we're going to have the postsynaptic side. So this is our muscle cell over here. We have our receptors on the postsynaptic side, which acetylcholine is going to bind to, and ultimately here is the end plate. You may see end plate potential or synaptic cleft, neuromuscular junction. It's all the same thing, but when we talk about a neuromuscular junction, the synaptic cleft, or a motor end plate, this is what we're talking about right here. Okay, so let's talk about a motor unit. A motor unit consists of one alpha motor neuron and all of the fibers it innervates. If we want fine control, aka digital manipulation, or to move our eyes up and down, there will be a small number of cells being activated because that gives us more control. So it's considered fine motor control. The number of cells with one motor neuron that innervates the glutes can be thousands. A motor unit is the most basic aspect of skeletal muscle function. So gross control, so postural muscles or using for example, our gluteal muscles, one alpha motor neuron could innervate thousands of muscle fibers, just controlled by one alpha motor neuron, which is right here. However, if we want to do fine motor control, like use our fingers or move our eyes up and down or to the side, one of these motor neurons may only innervate 5 or 6 muscle fibers, because that's going to give us a lot more control, and that's what we want with our eyes and our fingers and so forth. But for example, gross movement or gross control, one of these motor neurons can innervate thousands of muscle cells, because that's working on posture, stability, and gross movements like propelling ourselves up the stairs and so forth. Okay, so with transmembrane voltage, the characteristic of excitability is determined by the existence of this transmembrane voltage. This voltage is determined by 2 things. The sodium-potassium exchange pump is number 1, and 2, the difference in permeability between sodium and potassium. The cell membrane is 50 to 100 times more permeable to potassium than to sodium. The charge that is generated is an electrochemical charge, and if that charge is going to depolarize and initiate an action potential, the electrical current that allows that to happen will always flow in a complete loop. I'll say that again. The charge that is generated is what's called an electrochemical charge. If that charge is going to depolarize and initiate a signal, or the action potential, which is the electrical current that allows that to happen, will always flow in a complete loop. Okay, so here's what I'm talking about. We have the inside of the cell on this side, and we have the outside of the cell, so extracellular space out here. Here is our membrane, our plasma membrane, and then here's the inside of the cell. I do like this diagram because it shows that there's a high concentration of sodium on the outside of the cell and a little amount of potassium on the outside of the cell. And then it flip-flops. On the inside of the cell, we have a high amount of potassium and little amounts of sodium. But what this diagram doesn't show is the negative aspects of it. So I'm going to try to draw something for you to show that. But ultimately, what's happening here is when we get hit with an electrical signal coming from the nervous system to the cell, we're going to alter the permeability to this plasma membrane, and we're going to change the inside negativity to positive. Resting membrane potential in skeletal muscle tissue is negative 70 millivolts. So on the inside here at rest, it's negative 70 millivolts. So the inside of the cell is negative, and that's considered a polarized state. Okay, so I'm going to do my best to draw a cell for you here. Okay, so here we have the phospholipid layer over, here's our cell, and then here's this is going to be the inside of our cell. This is going to be the outside of our cell right here. So we are going to have a large amount of sodium on the outside of the cell and a little amount of potassium, and we're going to also have a large amount of the negative chloride ions outside the cell. Now moving to the inside of the cell, we are going to flip-flop. We're going to have a large amount of potassium on the inside of the cell and a very small amount of sodium on the inside of the cell. But I just explained that the cell resting membrane potential is negative 70 millivolts at rest before it's hit with an electrochemical signal for it to depolarize. Why is it negative? Right? So because what happens here is we have a large amount of negative anions on the inside of the cell. I just explained how potassium, this membrane right here, is 50 to 100 times more permeable to potassium than it is to sodium. So we are going to have a lot of potassium going through potassium channels on the outside of the cell. What's going to happen is we're going to leave these negatively charged anions behind, and we are going to, that's what gives us our resting membrane potential of negative 70 millivolts. And don't forget, the sodium-potassium pump is working via active transport as well. So for every 3 sodiums we move 2 potassiums back into the cell. So we're still moving 3 sodiums out, going against its gradient, and we're bringing in 2 potassiums. But since we're bringing out something that's 3 positive versus 2, we're still -1, so that even leads further to the inside negativity of the cell, which gives it the negative 70 millivolts. Okay, so when we talk about that bilipid layer of phospholipids, in many places there are integral proteins embedded in the bilipid layer, and these serve 2 purposes. So where are these integral proteins? These are them. These brown filled channels here, these brown structures on here. Okay, these serve 2 purposes. One, they make aqueous filled channels, which are water-filled channels, and are selective to what they allow to pass through. Some channels only allow sodium to pass through, and some channels only allow potassium to pass through. So this may be right here, this is a sodium channel. Right here is a potassium channel. They will limit large molecules passing through. The largest molecule that actually really passes through these is a glucose molecule, actually. Now, and number 2, the intracellular environment is regulated by these proteins. So these proteins are selective with what they allow in and out. We have some sodium channels, we have some potassium channels, and then also the intracellular environment is regulated ultimately by those, because again, we let 50 to 100 potassiums out of the cell for every 1 sodium coming in the cell. We're leaving behind those negatively charged anions on the inside of the cell right here. That's why resting membrane potential is a negative number. It's negative 70. And for a fun fact, not that you need to know this for any reason, but this lipid layer, just to give you an idea of just how small cells actually are and how thick this layer is, it's a biphospholipid layer. It's 75 angstroms thick. An angstrom is 100 millionth of a centimeter thick. So it's pretty small.